The last of the four carbon-based molecules of life is known as a nucleic acid. So as I'm sure you're aware by this point, all of these carbon-based molecules of life are polymers of some type of monomer. So the monomer for a nucleic acid is known as a nucleotide. Okay, so nucleic acid, nucleotide. There are four different types of nucleotides. Okay, you can see their abbreviations here. You have A, T, G, and C. So the four different types of nucleotides are based on their bases, which we'll talk about in just a second. There are four different types, so they are adenine, guanine, cytosine, and thymine. So those are the four major types. Thymine does eventually get replaced with uracil, but we'll talk about that when we start talking about RNA. So nucleic acids come in two different forms. You have DNA or RNA. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. So nucleotides have three different parts. Okay, you have your sugar, which is either a ribose or a deoxyribose. Only difference is that ribose has an extra oxygen, hence the deoxy, okay, deoxygenated, fewer oxygens, okay, in the deoxyribose. The major difference between these two is that ribose is a lot less stable. So it degrades much faster than deoxyribose. Okay, that extra oxygen is kind of like an attachment site for all different kinds of enzymes and compounds to break up the RNA. So DNA is actually a lot more stable, especially long term, than RNA. Okay, hence why DNA is our genetic storage material, while RNA is only used temporarily in the cells. You also have a phosphate group, so this is going back to when we were talking about the lipids and that phosphate group. Same thing here, okay, it's the PO4 3-. And you also have your nitrogen base, which are the four different bases that we talked about on the last slide, the A, T, G, and C. So this is the structure that you get here. Okay, you have your phosphate group. It will stick out on the outside of the molecule, so DNA is negatively charged because of this phosphate group. And then the phosphate group will be bonded to your sugar here, either the deoxyribose or the ribose. That extra oxygen on the ribose would stick out right here. So again, it's really at a great place to be on the outside of the molecule where it can get kind of attacked the easiest. You'll also notice that even though the sugars we talked about in the last section were mostly 6-ringed sugars, this one here is a 5-ring. Okay, just a little interesting thing to note. And then you also have your nitrogen-containing base. So these are the A, T, G, and C's that we looked at on the last slide. Two of them, we'll take a look here. Okay, so two of them have two different rings. So your adenine and your guanosine have two rings. Your guanine, adenine and guanine, have two rings, two ring structures. And then your thymine and cytosine only have a single ring structure. You'll also notice that it's a 2 rings bonded with a 1 ring and a 2 rings bonded with a 1 ring. Okay. All right. So nucleic acids store or transport genetic information in the cell. For humans, the genetic information storage system is DNA, and then the transport is RNA. For some bacteria, you will only have RNA. Some viruses only have RNA. Some will also have DNA involved, but yeah. So again, DNA is your deoxyribonucleic acid, which stores your genetic information. Okay. It is double-stranded. Okay, so it forms a nice little spiral. It's called the DNA helix. Okay, this is showing DNA replication, or actually this is creating RNA, but we'll again get into that later. I know I'm saying that a lot, I'm sorry, but then you also have your RNA, which is ribonucleic acid. So it transports genetic information around the cell in a human cell and is also responsible for basically copying the genetic information out of the DNA and then taking it out into the rest of the cell where that information can be used to create proteins, which then will go on and complete the function that the DNA is coding for. Okay, so this is your RNA. RNA is the single-stranded molecule here. Okay, again, it is less stable in the cell, so it can last anywhere from maybe a couple minutes to a couple hours, depending on the structure of the RNA itself. And that is kind of used as a fail-safe in the cell. So the RNA, if it's created incorrectly or if it's got an incorrect sequence, that's okay because it's going to get destroyed pretty soon. So when the cell is making RNA versus making DNA, with RNA it's going for speed, with DNA it's going for accuracy. Okay. Alright, so just a reminder, your 4 carbon-based molecules of life are carbohydrates, lipids, proteins, and nucleic acids. Okay, so these two sections, 2.3 and 2.4, really go hand in hand. So some of the videos for this section actually deal with all 4 of these molecules. Okay, and it's just kind of going back and reviewing all of it, but they all go really hand in hand. But yeah, so that is your 4 carbon-based molecules of life.